This investigation was on antimicrobial effects of Paeonia wendelbois extracts against Escherichia coli, carried out by Elena Saghafi, from Department of Biology, Dubai International Academy, Dubai, United Arab Emirates.
Hello everyone. So first I would like to extend my gratitude to Professor Ahmadi for all his support and guidance in providing this opportunity for us all. Peony wendelbois, commonly known as Wendelbois peony, is a species of flowering plant native to the Middle East, specifically Iran and Turkey. The flower typically blooms in late spring to early summer, and it has been traditionally used in medicine, and the effects have always been studied. This flower contains phytoestrogen, which can act as estrogen in our bodies, and it can influence hormonal activity. It has also been used to reduce depression, anxiety, and it has been supported by many studies throughout the years. This studies supported that the flower can affect the serotonin levels, which will result in the reduction of anxiety and depression. The compounds in the root may regulate the immune system and are used to treat conditions like rheumatoid arthritis and psoriasis. The peony extract may improve blood flow, reduce pain and swelling, and has been used to reduce menstrual cramps or any other inflammatory conditions. The antibacterial effect of peony species has always been studied. Well, peony wendelbois hasn't been studied specifically, but the related species has always been a center for research. In this paper, we're studying the antibacterial effect of peony wendelbois against Escherichia coli specifically. Escherichia coli, often abbreviated as E. coli, is a versatile bacterium commonly found in the intestine of humans and animals. Many strains of E. coli can be harmless or even known as beneficial to human body or animals, while others can be very harmful and cause diseases. These diseases can vary in intensity, so they can cause simple fevers, or they can cause diarrhea, often bloody, and inflammation. The aim of this study, we wanted to examine the antibacterial effect of peony extract on Escherichia coli growth, which was assessed in bacterial culture medium. For our methods, we first collected the plants, and it was preserved with care to protect its botanical integrity. The flowers that we chose were fresh flowers. They were dried, and first they were shaken to reduce any debris or dirt that was in excess on them. They were hung upside down and in a dark room to dry. Once the flowers were completely dried, they were stored in a cool, dry location to avoid any moisture and any contamination for the flower. The antibacterial of the leaf, stem, the root, and the flower was assessed during this experiment. E. coli culture was prepared in the appropriate growth medium, and the peony extract was diluted to reach our specific and desired concentration. The culture medium for E. coli was inoculated with a bacterium, and the varying concentrations of peony that we had was added to the two separate culture mediums. The culture mediums were then incubated in the optimal temperature for E. coli growth, which is typically between 37 to 42 degrees Celsius. After the designated time for the incubation period was done, the clear diameter around the marked parts that was where we had put the plant extract were assessed, and the antibacterial effect was seen in those parts. So our results showed that the peony extract did in fact have antibacterial effect on E. coli, and right now further research is being conducted to get better results. Our findings suggest that the flower possesses antibacterial effect, and different parts of it have different effects to different intensities, with the stem being the best part for the antibacterial effect, and the leaf having the lowest percentage of the effect. Thank you.
Okay, as I know, and you know, of course, we have not many recourse about the antibacterial effects of this plant, okay? And so, what do you know about the, location of growth, geographical location of growth of this plant? In which geographical places we can find this plant?
So, as I mentioned, it's native to the Middle East, specifically Iran, and it's found in more in parts where the weather is cooler, like mountain parts, and specifically, for example, in the north, and also in Turkey.
In Turkey, yes. And as I know, you are working in this research, okay? And do you think that you will get different results when you use different parts of the plant? For example, the flower extract, the stem extract, as well as root extract. Do you think that we will get different results about the antibacterial effects on E. coli?
Yes, there was a slight difference in the experiment that we conducted, and the leaf had the lowest antibacterial effect. It was 4 MCI, and the stem had the highest. It was about 32.
Mm-hmm. And this is about the compounds, because the compounds or bioactive material that are found in flowers, stem, and root, they are different. And this is interesting to say that if we use the plants from different places, however, in the same geographical part, again, we will get different compounds and different results. Okay, thank you very much, Elena. It was a very great speech. Thank you.